let's see where we're at with DNA polymerase. Replicates DNA in which polarity? Come on. Yep, adds new nucleotides to the 3' end of the growing chain. So if you have, as we saw in PCR, here's a template strand, 5' to 3'. So it's a single-stranded molecule of DNA. If we're going to draw a primer in here, I'll draw it in the middle here just for fun. Which end, this is what the question was asking, which end of this molecule does replication proceed in? What's the molecule, what's the atom at the 5' end of DNA? Think about an A nucleotide. 5' carbons attached to, normally the 5' carbon, we've looked at these before, is on the top left. There's a phosphate. So there's a 5' phosphate, and at the other end, the 3' end of every nucleotide is oxygen, hydroxyl, and OH. And we know that this primer is anti-parallel, 5'. There's a phosphate there, and there's 3'. And polymerase starts at the 3' end of the primer and moves to the left. So it's adding nucleotides to the 3' end. Of a strand. This, unfortunately, is one of those things you do just have to memorize. As you saw on Friday, you can look things up during the exam, but this is one of those things that if you're going to memorize anything, memorize this. 3' end is where DNA synthesis occurs. And so the second question on the Socrative quiz was exactly the same question. So I bet if you got this one right, you probably got the other one right as well. Which direction does DNA polymerase move? Ooh, interesting. We've got split decision. Let's go back. So is RNA polymerase moving towards the 5' end of the template or the 3' end of the template? Well, it's already drawn up here. Here's the template strand. So if it's adding on to the 3' end and moving this direction, it's moving towards the 5' end of the opposite strand. This is a fact that just comes out of the fact that DNA is anti-parallel. If it's adding nucleotides onto the 3' end of a strand, that means on the opposite strand, it's moving in the 5' direction. So it was just I was being unnecessarily tricky. Same, two questions basically asking about the same concept.
So will you always ask whether you want the original, in terms of the direction of the original strand, or are you going to ask for the new developing strand?
Either. So that's the only thing you need to be able to distinguish those two is knowing that DNA is anti-parallel.
So if your question asks which way does it grow in terms of the new developing strand, we would still say 3' to 5'?
DNA polymerase adds nucleotides to the 3' end of a strand. In other words, here's a way to think about this. Here's a double helix, or at least there's a double helix. There's a telomere. There's another telomere. This is overly simplified, but I think it maybe is a good way to answer this question. Label the polarity. So we've got 4 ends of 2 strands. So you should be able to put 2 5' and 2 3' on this molecule. Where's DNA polymerase? If this is a molecule that's being replicated. It's on the top strand, sitting right there, adding nucleotides. So polymerase is moving in that direction. If polymerase is there, which prime is that nucleotide that's sitting on top of? It's the 3' end of a growing strand. So that means that we can then fill in the other three polarities. So that's all you need. You only need one piece of information to answer any question about DNA and polymerase and polarity. If you get one of the polarities, one of the 3' or one of the 5', you can fill in the other 3. And I'll talk about the growing strand, the top strand in this case, or the template strand, which is the bottom strand.
So on the original template strand, it's moving in the 5' direction.
Towards the 5', or you could say away from the 3'. That's the direction polymerase is moving on the bottom. It's always, though, the only thing you have to remember is, my point is that it adds to the 3' end of the growing newly synthesized strand. After you know that piece of information, then you can figure out the polarity of all of the other carbons in a double helix.